Okay, so we're back with dihybrid crosses. Others, other of Mendel's experiments had him looking at two separate traits, so two separate characteristics. So, for example, he looked at seed color and seed shape at the same time. When he did that, that's what's called a dihybrid cross. You're crossing two different, which is di, okay, two different traits. So here's a picture example. We looked at true breeding yellow round peas with true breeding green wrinkled peas. Okay, so when you cross those two, the F1 generation ended up being 100% yellow round peas. That's because yellow and round are the dominant characteristics. Okay, so that would mean green and wrinkled are going to be recessive. So then the F2 generation is then going to produce different percentages of each type. So we would have a 9 to 3 to 3 to 1 ratio, which means 9 out of the total would be yellow round, 3 would be green round, 3 would be yellow wrinkled, and 1 would be green wrinkled. Okay, so I'll show you how to do the Punnett square for that in a little bit. Okay, so two organisms can have the same phenotype, but they can have different genotypes. So if, for example, if you're looking at homozygous dominant versus heterozygous, so to determine the genotype of an individual with a dominant phenotype, you have to do the Punnett square. Okay, so you breed the dominant phenotype and the unknown genotype with a homozygous recessive to determine the identity of the unknown allele. Okay. So let me. Okay, so here's another practice. So watch the other video showing how I'm breaking down our genetics practice questions, but go ahead and read through the questions and try them out on your own. So, Mendel's first law of heredity is the law of segregation. What this means is that during meiosis, the alleles segregate. What that means is homologous chromosomes separate, and each allele for a trait is packed into a separate gamete. Okay, so that's why we get half of our traits from our mom, half of our traits from our dad. So if you look here, you can see we have, oh, go back. Okay, we can see that, why is this doing it? Okay, so we can see the line drawn here in between the two chromosomes, okay? So what they're going to do is they're going to separate, okay, within each single gamete. So that way you can get the genetic variation. Okay, so here's more explaining the law of segregation. So if you look at, this is still all in meiosis because these are all of our sex cells because of getting our genetics from our parents, okay? So we have a homologous pair of chromosomes. Okay, and then we go through anaphase 1. Once we go through anaphase 1, that's where we can get the segregation happening. Okay, so his second law of heredity is independent assortment. Different genes separate into gametes independently. So our genes decide on their own where they're going to go, how they're going to get split up, and in which gametes they're going to be in. Okay? So your non-homologous, so not same or similar chromosomes, align independently. So if we're talking about yellow versus round, yellow versus green and round versus wrinkled, okay? So different classes of gametes are produced in equal amounts. Sorry, Trey accidentally started playing music. Okay, so different classes of gametes are produced in equal amounts. But this is only true for genes on separate chromosomes or on the same chromosome. But they're so far apart that crossing over happens very frequently. So if you look here, we have our both heterozygous dominant traits here. Okay, they can get split up in all of these different fashions. Okay, so we can randomly and independently assort them in different ways. And that's how we get all of the genetic variation. So, which stage of meiosis creates the law of independent assortment? That's going to be metaphase 1, okay, because that's when we can have a lot of the crossing over happening. The only exception is if genes are on the same chromosome and are close together. Those are usually going to be inherited together just because they are so close to one another. They're kind of linked, so they rarely cross over separately. So here's another practice problem. Go ahead and pause, try it out on your own, and then watch the video of me breaking it down to see if you got it correct. Another practice. Okay, so here's just a quick review of the 2 laws of heredity. So we have law of segregation in anaphase 1 and the law of independent assortment in metaphase 1. Okay, so beyond Mendel's laws of inheritance. Okay, so we're going to kind of take this next step forward, and then we'll go over some genetic disorders. So he worked with a very simple system, okay, because P's are very genetically simple. Most traits are controlled by a single gene. So for example, I have brown eyes, and that's by that one single gene of eye color, okay? But each gene only has 2 alleles, okay? So one of which is completely dominant to the other. So I got one of those alleles from my mom, one of those alleles from my dad, and those are each dominant to the other. Okay, so the relationship between genotype and phenotype is rarely that simple, okay? So a lot of, and most genetics are more complicated, okay? So for an example, we can have something called incomplete dominance. That means that a heterozygote shows a kind of intermediate or blended phenotype. So it's like in the middle. So an example, Miss Salyer's breaking things in my house. Sorry. So you could have red flowers mixed with white flowers, okay? Now you can get incomplete dominance, which would then lead to pink flowers, okay? They make 50% less color, so that means the protein expression is altered. Okay, so if the protein changes shape, obviously what else is going to change, guys? You should all say changes function. Perfect. Oh my God, I know you miss me. Okay, so when this happens, you can kind of get a mixture in color or whatever trait it is in the phenotype specifically, because one doesn't fully dominate over the other. It's incomplete dominance. So then you can have something called codominant. So 2 alleles affect the phenotype equally and separately. So it's not a blended phenotype. It's going to be both are very, very present. Okay, so an example would be the human blood groups. Okay, because you can have 3 different alleles. Can you stop breaking stuff? You can have IA, okay, so that's just the I is representing what that letter would be, and the A and the B are going to be representing the blood type. Okay, so you can have A and B alleles are codominant. Okay, that's why you can have type A blood, type B blood, or you can have type AB. Now, that has to do with the glycoprotein antigens on the red blood cell. So what they can actually monitor and read. Okay, so if IA and IB, both antigens are produced, then you would have the blood type AB. Okay, so this is kind of to give you an example. The lowercase I just represents like nothing, kind of, if that makes sense. So your genotype for your phenotype of type A blood, this is what it would look like. So you would have type A antigens only. So antigens are the little receptors, okay, on your surface of your red blood cell. You would have anti-B antibodies, meaning your immune cells would attack type B blood. You can have type B blood. This would be your genotype. That would mean you would have type B antigens, and you would have anti-A antibodies. Now, type AB, that's our codominant, okay? You would have no antibodies, okay, because you can accept both of them. These are going to be your universal recipients. Type AB blood can receive blood from anyone. because they don't have any antibodies. Type O blood is where you are kind of considered, I guess, the recessive, you would call it, where it's just the 2 lowercase i's for your genotype. You have no antigens. So since you have no antigens, that means that you are the universal donor. You can only accept type O blood because you have anti-A and anti-B, but you can give blood to anyone because you have no antigens, so you don't read any specific blood type. So here's another example of genetics practice. So go ahead on a piece of paper, do a Punnett square and everything for blood type, and then check the video to see if you got it right. Another example. Okay, so let's get into diseases and disorders. You have pleiotropy. Most genes are pleiotropic, which means one gene affects more than one phenotypic characteristic. So one gene affects more than one trait. So this gene can affect multiple traits. So for example, for pleiotropy, that usually results in dwarfism, which can be called achondroplasia, or giantism, which can be called acromegaly. Okay, so pleiotropy is where you have this one gene that ends up affecting more than one trait. So if I have the gene for eye color, it could affect more than just the trait of eye color. It can affect all sorts of things. Okay, so these are pictures of the dwarfism. Okay, and then the next slide is going to be the pictures of giantism. Okay, there's this famous guy, he did end up dying, named Andre the Giant. Okay, he was massive, but when you are, so huge or so small for dwarfism, your life expectancy is going to be shorter, because imagine if you're so small, all of your body organs being cramped in such a tiny space. But then if you're so huge, imagine your heart working so hard to pump blood out into your body. Okay, so your organs have to kind of go into overdrive to make your body function. So here is a lethal mutation for dwarfism. Okay. So let's say you had a dwarf mixed with a normal person, okay, and then 2 dwarfs mixed together. We have dominant inheritance. So we could have 50% dwarf, 50% normal. Then if we mixed a dwarf with a dwarf, that's where we could get a lethal version, okay? That would be the homozygous dominant, okay? That is lethal. It will kill you. That's just basically too much of the dwarfism traits for your body to even function. We can have something called epistasis, where one gene completely masks another gene. So not where it masks the allele, but it masks the gene entirely. Okay, so for example, that's coat color in mice. You have 2 separate genes for it. Okay, you can have uppercase C, lowercase c, where it's pigment or no pigment. You can have uppercase b, lowercase b, where it's more pigment or less pigment. So more would be black, less would be brown. But then if you have lowercase c's for both of them, it's going to end up being where it's homozygous recessive. That's where you become albino, okay? It doesn't matter whether your B allele is dominant or recessive. You're going to be albino if you have homozygous recessive for pigment. So in laboratory retrievers, oh my God, look, it's dogs. Yay. Okay, so if you have no dark pigment in your fur, okay, you're going to have your recessives. But if you have dark pigment in your fur, that's where you're going to get chocolate and black labs. Okay, if there's no dark pigment in the fur at all, you're going to get yellow labs. And this even affects their nose color, their lip color, and the rims of their eyes. I'm going to pause and then we can pick it up in the next one.